Hi there, and welcome back to the channel. I'm the GCSE science teacher, and in today's video, we are learning about enzymes. This is for GCSE biology students. If you do find this helpful, feel free to like, comment, subscribe. It'd be great for you to join our community. And thank you so much to everyone who already has. It's great to have so many subscribers in such a short period of time. I'm very fortunate. So thank you. I'm really excited to build the community together. If you are also looking for more help and support with GCSE physics and chemistry, feel free to check out my Instagram and TikTok, which is at the GCSE science teacher as well, where I share lots of helpful tips daily on TikTok and regularly on Instagram. So, the topic, like I said, is enzymes, and this links to GCSE chemistry too. So, if you are studying that, this should be helpful towards those studies as well. So, first of all, what is an enzyme? Well, it's a biological catalyst, and like you know in chemistry, a catalyst is something which speeds up the rate of a reaction without getting used up. It does that by lowering the activation energy or the amount of energy needed for a reaction to happen. And enzymes are kind of like a catalyst because they actually increase the rate at which a reaction happens. And when we talk about reactions, we're talking about biological ones. So it could be a specific reaction to help with digestion and things like this. They are also proteins. So they're made of amino acids. They are 3D in shape, and that is specifically important so that we understand that because they're 3D, they have a very specific shape. And this is specific to the enzyme. And that links to other places in the curriculum as well. If you've been learning about the immune system, you'll know that antibodies and antigens, they are complementary to one another. They are specific, and only certain antibodies will bind to certain antigens. And it's the same with enzymes. They have a very specific shape, and that is how they actually work. I'm going to talk about that today. But enzymes are actually all around us, not just in the human body, but in other animals as well. We also use enzymes in the food industry. So if you've ever eaten cheese or you've ever seen it in a shop, I'm sure you have, you'll know actually that cheese is made from a specific enzyme called rennet, which actually comes from cows. This is an enzyme that helps milk to curdle and forms that cheese that we can buy and consume. Interestingly, as well, linking to this, some people are lactose intolerant. This means that they can't break down or digest a certain type of sugar that's naturally occurring in milk, which is called lactose. So, lots of people who have a lactose intolerance, it's because they don't or haven't got enough production of an enzyme called lactase, and that helps break down that lactose into more digestible parts. So often those people will suffer with stomach pain or stomach cramps, and they'll have to have different types of dairy products to get that particular nutrient, which I thought was quite an interesting one. So, when we talk about enzymes, a lot of the time we link it to this idea of a lock and key theory or a hypothesis. And this is just an idea that scientists have used to kind of explain how something works. And as you can see, a hypothesis is our best idea. This is a theory. We haven't really got any evidence to say why this wouldn't work. It makes a lot of sense that it does. If we find new information, we'll update this with time. It's kind of like the atom, how that's changed over time. It's the same with any model or theory in science. Until you get more evidence, you kind of go with what you have, right? So, a lock and key theory, or the hypothesis of it, says that an enzyme is like a lock. That's the analogy, okay? And the substrate or the molecule that fits into the enzyme is like a key. And that means that it's specific to the lock. So, it's all about specificity, it's all about complementary shapes and how this substrate fits into the enzyme. Now, in the enzyme, you have a very specific area called the active site. And this is where the substrate or the key would fit to open the lock or to activate that reaction. And the active site is the most important part of the enzyme because it actually allows the substrate to fit into it. But there is a misconception here, and I find this amongst students. Sometimes students say that substrates or the molecule that fits into the enzyme is the same shape as the enzyme's active site. What they actually mean, and what you should say in your exam, is the substrate is a complementary shape to the enzyme's active site. That just means that it can fit into it because it will actually fit and combine nicely and bind well. And we want to say that when the enzyme and substrate actually do combine, we say this is now an enzyme substrate complex which has formed. So, some of the examples of enzymes that you may have seen across the curriculum, and you may have seen quite a lot actually, are the following. So, you've got amylase, protease, lipase, carbohydrates, DNA polymerase. blah, 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 blah. There's lots of different enzymes you can see there. A lot of them will end in the same suffix or the end part of the word, okay? They'll all end in A's, except for a couple. There's ones like pepsin, trypsin, which are part of the digestive system, but they don't have that A's bit at the end. But kind of like a hard and fast rule, not a particularly not the best rule because there's exceptions to it, but a lot of the time enzymes end in A's. So that's a bit of a tip to help you in the exam if you get stuck. Some of the key enzymes that you do need to be aware of are the following. So amylase, because it comes up in a required practical. Amylase is used to break down starch into a sugar such as glucose, and that's done in the required practical, yes, but also in the digestive system. So it links to different areas of the curriculum. Protease is another enzyme that comes up a lot. And this is an enzyme which breaks down proteins into amino acids. Lipase breaks down lipids into fatty acids and glycerols. And protease and lipase, they kind of sound like what they're breaking down. So it's quite nice. Remember starch. I know I talked about the digestive system, but remember starch, we consume starch through the foods that we eat. We don't store starch, we store glucose as glycogen. That's one of the misconceptions that I thought I'd mention here as well. So at GCSE, they like you to talk about the factors that can actually affect how well enzymes work and how easily they can catalyze reactions. So the things that can affect enzymes are the temperature, the pH, and the substrate concentration. I'm going to talk about each of those in a moment. But remember that enzymes, because they're biological catalysts, they are able to perform well at specific conditions. And this depends on the type of enzyme. Remember, we can find enzymes in the stomach that work well at a very low pH, a very acidic concentration of acid or a level of acid. You have enzymes that work really well in more alkaline conditions or less acidic or perhaps even neutral conditions. It just depends on the environment you're in. But the best way to kind of describe this is to say that enzymes work best at their optimum temperature or pH. And that just means it's the best level for them to work at. And if there's any deviation from this temperature or pH, it can actually slow down the rate of reaction or it can even stop it altogether. And we'll come on to why that is now. So if the reaction stops completely, it's because the enzyme has denatured. And this just means the enzyme's active site has no has now changed shape. Essentially, it's an irreversible reaction. We cannot get back to the pre denatured state. One of the key misconceptions here is that students will say enzymes have died or they have been killed. Even destroyed, I would avoid saying. I would go with the idea that the enzyme's active site has changed shape, the enzyme is denatured. That's going to get you the most marks because it's the most accurate. Remember, enzymes, although they are biological catalysts, they are not living, so they cannot die. And the two factors that can actually cause the enzyme to denature is the temperature change or pH. And specifically with temperature, you're talking at GCSE about an increased temperature as well. So remember, if the enzyme's denatured, that means the active site has changed shape. This is irreversible, and the enzyme can no longer form enzyme substrate complexes because the substrate is no longer complementary to that enzyme's active site, and the rate of reaction slows and stops altogether. So, one way they could ask you how to kind of describe this or explain it or just to find some information about it or infer something from it is by looking at graphical data. So here on the side you can see that the rate of enzyme activity across temperature, you can see that at the beginning, if you increase the temperature, the rate of enzyme activity is pretty good. It's increasing as well. And that's because if you increase the temperature, you're increasing the amount of available energy in terms of thermal energy and kinetic energy provided to the substrates. And that means that they're able to collide more frequently with the enzyme's active site. And therefore, the rate of activity of the enzyme increases. And then we get an optimal level where the enzyme is working at its best. It's living its best life, it's doing its thing. But then all of a sudden, if you increase it higher than this, the enzyme active site changes, it denatures, and therefore the substrate can no longer fit into it. It's no longer complementary, and the rate of reaction stops altogether. You can see that there's quite a strong decrease there. It's the same with pH. However, remember the pH could be very acidic or very alkaline or very neutral. It just depends. But there will be some denaturing going on if the enzyme is not at the right pH. Substrate concentration is slightly different. You won't get denaturing going on here. What's happening is, let's say we had 10 enzymes, but only let's say we had 10 enzymes and 100 substrates, you can only fit 10 substrates into 10 enzymes at one time. And that means that the enzyme is a limiting factor. So if you increase the substrate concentration and you get to a saturation point where you've gone to 11 substrates, you've gone to 12, 13, 100, you can only get the same level rate of enzyme activity because there's only so many enzymes that are there. So you get to this point where it's like the enzyme can't do any more. It's going as fast as it can, and there is a saturation level. So that's one way they could ask you about enzymes and graphs. I hope you did find that helpful. I've been the GCSE science teacher and you have been curious. Thank you so much for your time. If you did find it helpful, feel free to like, comment, subscribe, join our community here on YouTube. Also, check me out on Instagram and TikTok for more helpful content for GCSE biology, chemistry, and physics. And in the meantime, have a great day and I'll catch you in the next one.